so much. It's really wonderful to see so many people show up, and it's always very humbling to speak in front of somebody who's like lived with some of the medical problems that we're trying to treat.
Is your mic on?
I'm not sure. Can people hear me okay?
There's a switch. There's a switch on.
It's on. I turned it on. Hello? Can you hear me? Okay, there we go. Good. Okay, anyway, I'll just start by thanking again and saying how wonderful it is to be here at the Koch Institute and how nice it is to see everybody here tonight. And what I'd like to sort of share with you tonight is sort of a little bit of my perspective on how what I think is actually, having lived part of the story, it's kind of easy to get tied up in the science and some of the particulars and the controversies the scientists argue about. But I think, hopefully, as you'll see over the course of the evening, this really is an amazing story that, sort of culminating in sort of the humbling process of talking to somebody who has actually lived this and hopefully won't need to benefit from anything that we're talking about, but, if necessary, he might be able to. And so I think it's well accepted that in the scientific community and well known that metabolism is altered in cancer. Arguably, changes in metabolism were one of the first differences noted between cancer and normal cells. This dates back to the 1920s, before people even cared about, metabolism or really studied cancer. And, I think if we fast forward now almost 100 years and asks, what have we learned from these sort of changes in metabolism? I think from a clinical perspective, much of it has to do with FDG PET scanning. And as anyone who's ever been, had an ordeal with cancer or has, a loved one who's been touched by cancer probably is familiar with this test, which really measures the altered glucose uptake of cancer. And so a radioisotope, fluorodeoxyglucose, that's what the FDG stands for, which is taken up and trapped into cells and can be read out on this positron emitting scanner, really can use this test as a way to identify and stage different kinds of cancer. Now, what has become clear with the use of FDG PET scanning is that as it's come to wider and wider use, it's been understood that it actually can also be quite predictive for how well patients will do if they are treated for whatever kind of cancer. So here's two examples of that. This is a patient with a sarcoma. That's a tumor of the soft tissue. It's a gastrointestinal stromal tumor, which have a specific mutation that's sensitive to this drug. And this patient here has tumors in the liver, and he was treated with this drug. And this scan was done a couple days after the drug was started, and you can see that the tumor is on PET negative. And this sort of read out that this tumor had a response, and this patient went on to do fairly well on this treatment. And you can kind of know early on that this was a good drug for this patient, and that can make a real difference in their life. Here's sort of a patient with lymphoma. Their lymphoma is treated with, sort of classic chemotherapy, a regimen called CHOP-R, and it's exactly the same story here. The ability of this regimen to make the tumor go PET negative really ended up being predictive that this patient would ultimately do well. And so this kind of has led many people, this type of use, well, this is useful for the tumors that are FDG PET positive and can help you know if drugs that we already have might or might not be working. It might suggest something different, and that sort of raised in many people's minds the idea, well, if this is such a good predictive marker of how cancers will respond, and metabolism is clearly important to tumors, we've known that for 100 years, maybe there's some way we could take advantage of these differences in metabolism to actually come up with better therapies. And, maybe if we just look back from history, this is not such a bad idea. In fact, the history of chemotherapy, if we go back to the earliest days, it's really about metabolism in one way or another. So this is Sidney Farber. He's one of the first people to give chemotherapy. Some of the first chemotherapy that was ever given was work he done giving to children at Boston Children's Hospital in the 1940s and 1950s. And the drugs that he was giving, although at the time they were given, people didn't really understand what they were doing, but it was actually this drug right here, something called aminopterin. The modern version is this drug, methotrexate, still used in the clinic today. And basically, there were amazing responses. For the first time, you could actually treat cancers in people. Diseases that they would send children home and, basically we don't have anything to do for you, suddenly had responses to these drugs. And later it was found out these drugs targeted metabolism. Now, as we moved forward in time, we sort of got interested in other pathways and other places, and it was sort of a few years ago that it was kind of realized that, well, maybe we should go back and think about metabolism as a target again. And you know, many of you are probably familiar with some of the work to try to understand cancer genetics. There's excellent work here, other places around Boston, around the world. And many people have said, well, what are the mutations that cause cancer? And it turns out that there were several rare mutations that cause cancer, and those mutations were in metabolic enzymes, and that got people thinking again about maybe we should, you know, think about metabolism and what it might be doing in cancer. And what those mutations were mutations here in two enzymes in the TCA cycle, or this also called the citric acid cycle. If you took a college biochemistry class or a high school biochemistry class, if you're young enough, someone forces you to memorize sort of these metabolic pathways in cells. So these are a couple of the enzymes that someone forced you to memorize. And these enzymes, it turns out, are mutated in some very rare but inherited forms of cancer. And then a very interesting thing happened in 2008, and that was suddenly this effort had been done where we're going to sequence the genomes of many different people and many different cancers, the so-called TCGA, or Total Cancer Genome Atlas, and this has sort of been progressing through different tumor types. And one of the first ones that they finished was brain tumors. Glioma is the formal word for it. And when they sequenced the brain tumors, they found that another TCA cycle enzyme, IDH, isocitrate dehydrogenase, also had some mutations in it. Now I can tell you when this first came up, because I trained with a person, Craig Thompson, who's was sort of one of the pioneers in understanding metabolism in cancer. He's now president of Memorial Sloan Kettering, so he's someone in the news every now and then. And the person who discovered this was discovered at Johns Hopkins. Bert Vogelstein called up Craig and said, wow, we have mutations in a metabolic enzyme. Does that mean anything important? And Craig said, it might, you know. And so they decided to look into this more, and they found that it turned out there were lots of these mutations in IDH, and could we figure out what it might mean? And over the course of the last several years, efforts to sequence lots of tumors, and all of a sudden, oh wow, there's this mutation in this disease, we can now start looking broadly. And we find, well, it's not just glioma. It turns out it's very common in certain types of glioma. We'll talk more about that in a minute. But it's also pretty common in a bunch of other cancers, including some cancers that, you know, many people certainly aren't on the forefront of, you know, mainstream cancer research. And this got people thinking, well, you know, what does this really mean? And I think some of the data that really suggested this might be important actually was here in one of the very first publications about IDH mutations. And this is looking at patients who have an IDH mutant tumor, so have an IDH mutation by sequencing, versus those that don't, and just asking how do they do over time. And this is pretty obvious. This is basically how long do they survive. Doesn't say anything about how they were treated, just how long do they survive. And there's this huge difference in what goes on. Patients with an IDH mutation do better than patients that don't have an IDH mutation. Now, this particular human study, it confuses a lot of people. Because a lot of people come up and they say, oh, what is IDH mutation doing that's actually good for you, okay? In fact, I've had people call me up and say, shouldn't we be like using gene therapy to put IDH mutations in people's cancers? And I'm like, that is, first of all, that's insane, okay? But second of all, I think what this really tells you is that these are really different diseases, right? We tend to lump cancers by type. That's sort of the historical way that we thought about cancers. But as we've learned more in how, you know, the genetics and the molecular biology of tumors, we know that just because a tumor arises in the brain doesn't mean that every tumor that arises in the brain is exactly the same disease. In fact, it's probably many different diseases, and nice molecular work by other people has found that to be true. And it turns out this defines a different subset of disease with a specific natural history than this one. Now, why that's important to recognize is because it says that if we actually can understand that this is a specific disease, if we can identify the patients who have this disease, we can know that we should treat that disease as opposed to, you know, making therapies that, you know, really are targeted against 8 different diseases, because that plan probably is going to be less successful than if we can go after the one thing that we actually understand. So that in and of itself is already a really exciting finding. Now, it also suggests, well, maybe we can take advantage. If we can understand something about what is different, why this disease depends on these mutations, maybe there's some other pathways we might be able to go after. And, you know, kind of in support of that, lots of people have gone through and sort of studied this. And there's a slide from my colleague Dan Cahill at MGH, who has worked with us on some of the stuff we've done here at the Koch. And basically, what this slide illustrates is that it turns out IDH mutations are very early events in glioma. In the patients who end up getting this. And they know this is because oftentimes gliomas will grow out of like smaller, low-grade, sort of precancerous lesions. And those earliest lesions seem to have these IDH mutations. Now, this is also important because it says if you could take advantage of whatever, why this might be good for the cancer, it might actually be a good way to treat the tumor because in order to make the cancer at any time, at the very earliest stages, it seemed to require this mutation, arguing that it might be dependent on it forever, okay? Now, the problem is that if we go back to what's kind of raised this interest in cancer metabolism again with these loss of function mutations in TCA cycle enzymes, if it's the same phenomena that you lose the normal function of IDH, there's not a lot to target there. This is the tumor suppressor problem. If people are sort of more savvy in the audience and know about this, there's lots of proteins and genes that cause mutations that cause proteins not to work, and those prevent things that normally prevent cancer from growing, and that was sort of what people thought these were doing because they're loss of function mutations. It's hard to know how to replace a function with a drug, right? If you don't have something that doesn't work, if you have a protein that doesn't work, it's hard to know how to fix that. At least it's not easy to fix that. And so the hypothesis, at least when this came out, well, we know that these are loss of functions. Maybe these are also loss of function. So the experiment that someone does is said, well, let's see if this is really loss of function mutation. So isocitrate dehydrogenase is an enzyme. That's what IDH stands for. And it basically catalyzes this reaction. So I'm going to teach you guys a little chemistry, okay? So this molecule is isocitrate. It has 6 carbons, okay? This is the chemical structure. And it does 2 things in making, so IDH catalyzes this reaction. What someone made you memorize in your class in high school if you're young enough, or college or graduate school if you're older like me. What you do is someone taught you that IDH catalyzes this reaction to turn isocitrate into this molecule, alpha-ketoglutarate. Now, to do this chemical reaction, 2 things happen. So if you compare these 2 structures, you'll notice 2 things are different. One is that this group right here is gone. That's a CO2 group. Okay, it's gone. It's not here. And the other thing is that you've changed this hydroxyl group into a ketone. All right? So for the more chemistry inclined of you, this is a oxidation reduction reaction. That's what this NADH part is, and you lose CO2. So 6-carbon isocitrate to 5-carbon alpha-ketoglutarate. Every high school kid today can tell you that this is a reaction catalyzed by IDH. And so the obvious thing is that if these other things are loss of function, they will come back and say, well, let's measure this reaction and see if this is loss of function. And so you can do that. It's easy to set this up in the laboratory. And the idea is that because these other things have loss of function, let's find out if this is also loss of function. And so we do that experiment, and we can measure here by, sort of this NADPH fluoresces, and so you can measure the fluorescence of NADPH over time, and so this is what wild-type isocitrate dehydrogenase does. And if we make these mutants and test their enzyme activity, you can see that it catalyzes this reaction much less well, arguing that indeed this is loss of function, just like what was predicted might be the case. Now, this is kind of depressing if you're thinking about how could we take advantage of this, because this loss of function, how are we ever going to replace this function if we want to try to take advantage of this? Well, the real insight first came from thinking about, well, the genetics didn't really support this. So you don't have to be a molecular biologist to understand the idea. If you want to break your car, how many ways can you do that? If you want to make it so your car doesn't drive, you could, take the steering wheel off, you could remove the gas pedal, you could do 100 different things to make your car broken, okay? Yet, if you wanted to make your car go faster, you might only have one or two things that you might be able to do. And that's sort of the analogy of what happens here. And it turns out every single time there's an IDH mutation in glioma or in AML, it's in exactly the same residue. And in fact, it's in analogous residues between the two proteins. It's always an IDH1, R132, and an IDH2, it's always R172, which just doesn't fit with this idea that the enzyme is broken. But yet I just showed you an experiment that it doesn't work right. Well, this sort of requires going back and understanding a little something about enzymes. And it turns out the way we teach metabolism to people is that enzymes work in cycles. So the TCA cycle moves in this clockwise direction and always converts isocitrate to alpha-ketoglutarate. But of course, enzymes aren't that smart. Enzymes basically do chemistry and allow chemistry to happen. And it turns out enzymes really catalyze reactions in both directions no matter what your textbook says, okay? And so, and in fact, if you look in the literature, you can find evidence that IDH1 and IDH2, the two enzymes that are mutated, there's actually literature that they can work in both directions. And that's in contrast to this IDH3 enzyme, which catalyzes the same TCA cycle reaction that most people think about. And it turns out this is never mutated in cancer, okay? So that kind of screams out, well, the genetics say this is gain of function, it looks like it's loss of function in the direction everyone cares about, and the only two versions that are thought to be reversible are the ones that have this single point mutation. This really screams we should look at, does it have activity in the opposite direction? And so with much prodding and arguing among people, it actually ended up that we did the experiment and it turned out that it was informative, okay? So to do the experiment, you can take alpha-ketoglutarate and basically set up the exact same reaction, but see if it now, rather than makes NADPH, consumes NADPH, and the wild-type enzyme does not, and lo and behold, the mutant does, okay? So that's pretty cool, because it says now the mutant enzyme can do something that the wild-type enzyme can't. And now here's a little bit more chemistry, so you're gonna say, well, what can it do? Well, as I told you before, there's actually two things happen as you go from, between 6-carbon isocitrate and 5-carbon alpha-ketoglutarate, in Bill Kalin's terminology here, a 2-oxiglutarate, same molecule. Okay, so the two things happen. One is you have to take CO2 on and off, and the other one is you do this oxidation reduction reaction. Okay? Now, it turns out if we go in and look chemically, what does this mutation mean at the structure of the enzyme? Well, the residue that's mutated in IDH1 is this arginine 132. The residue that's mutated in IDH2 is this arginine 172. It turns out they're exactly the same residue. This is the catalytic part of the enzyme, so the part of the enzyme where the chemistry happens. And what both of those residues do is they actually coordinate that CO2 group coming on and off the molecule. All right? So remember, 5-carbon alpha-ketoglutarate, 6-carbon isocitrate. The difference is that carbon dioxide that I pointed out. This coordinates that coming on and off. And so smart people decided, figured out, sort of guessed that, well, maybe if you can't coordinate that CO2, you actually carry out the oxidation reduction reaction, but don't put the CO2 back on. And make this molecule, which no one had ever heard of or thought about at the time, 2-hydroxyglutarate. And it turns out if you do the experiment, I won't bore you with the details, that's exactly what you would get. So if you basically took alpha-ketoglutarate, put it through the IDH reaction, did oxidation reduction, but didn't re-add CO2, you'd get this molecule. It differs from alpha-ketoglutarate, ketone, 2-hydroxyl group. This is 2-hydroxyglutarate that's referred to as 2-HG. We could do a fancy mass spectrometry experiment to ask what's different between cells that have mutant IDH versus cells that have wild-type IDH. And it turns out there's 3 things that are different, labeled here A, B, and C. And if we go in and ask what is A, it is 2-hydroxyglutarate. And if you ask what B and C are, they're basically mass spec artifacts. So when you do mass spectrometry, you basically get different adducts come on to things, the chemistry that allows you to measure it, the mass spectrum. People who know about mass spectrometry know that this happens. And so they're really the same thing. The one thing that's different between these two cells is the production of this molecule. So that's pretty cool. So now we can go back and actually say, well, is this true in real patients? And so we can measure lots of metabolites in real patients. These are patient tumors that either have these are brain cancer patients, and it's looking at the tumor if you have a wild type or a mutant IDH. And you can see that most metabolites are the same. but there is a huge difference. This is a log scale. So there is 2 logs more 2-HG in the mutant tumors and in the wild type, saying that this really is meaningful. So this is pretty cool because now this suggests 2 things really important for this disease. And it says, if these are gain of function because you're making this metabolite that's not supposed to be there, 2-HG, it says 2 things. One, it says, well, they're gain of functions. If the 2-HG is important, maybe this is a drug target. If we can stop 2-HG from being made, maybe that's important. The other thing it says is that, well, it's making a metabolite that's not there. Is there a way that we might be able to use the presence of this metabolite to monitor the cancer? And so both of those are exciting possibilities. Both of those would be very useful. And Kate Yen, when she talks later, will tell you more about this. But basically, here's an experiment again from our collaborator, Bill Kalin, that demonstrates that IDH mutations, 2-HG is actually the causative agent. I won't take you through it for the sake of time. And if you look in AML, Kate might tell you more about this, asking what it might be doing. Well, the genetics really point to that it might be in the same pathway as this enzyme TETZ. I won't bore you with the details because Kate will tell you more about this. But it turns out that there's another family of enzymes out there that use alpha-ketoglutarate as part of there. There's, in fact, 60 of these enzymes in cells. Here's 3 different examples of them. They use alpha-ketoglutarate as part of the chemistry that they do to regulate gene expression in cells. And so what appears to be the case is that what this 2-HG does is it acts as a natural inhibitor of these many enzymes. And because these enzymes are so important for regulating gene expression, by having too much 2-HG, you basically screw up the way that these enzymes work. Gene expression gets messed up. Cells can't differentiate. You'll hear a lot more about that from Kate when she talks. Now, I want to spend a little bit of time talking about the monitoring. Kate will mention this as well, but I at least want to mention it. So monitoring is interesting. I showed you we can measure 2-HG by mass spectrometry. It's pretty easy. But if you have a brain tumor, it's hard to go in and take repeated biopsies, okay? You're going to hear from someone who's had this done. I'm sure he doesn't want his brain biopsied repeatedly to know if the drug's doing something, if it has an IDH mutation, et cetera. And so one idea is that because there's so much of this molecule here, could we use some non-invasive approach to look at that? And NMR spectroscopy is one way to do this, all right? So patients get MRIs. MRIs are part of the normal care of patients who have brain tumors. And it turns out anytime you do a clinical MRI, you show the pretty pictures of what the tumor looks like. But there's a lot more information in there. There's information about the metabolites that are present in the tumor, and that's read out here in this MR spectrum. And it turns out you can actually see 2-HG within that spectrum. It's kind of difficult to see. And a group at MGH, there's also another group at Texas that's worked on this. We've been collaborating very closely with the group at MGH, has figured out a way using clinical scanners, and I won't go into the details of how they do this because it's probably too much for even me most of the time, but people in this audience for sure, to really go in about how this actually works. But the bottom line is that they can actually go through and find 2-HG using this different MR spectroscopy. And maybe it's easiest to see here. So here's a patient who has an IDH mutant tumor. Here's a patient who has a wild type tumor. If we look here at normal brain shown in red, the tumor shown in blue, you can see that 2-HG is read out in the blue spectrum, but not the red spectrum, whereas you don't see it in this patient's tumor in blue that doesn't have an IDH mutation, but here in red. So you can actually genotype patients based on the presence of 2-HG using this type of technology. And we were funded through the MIT Bridge Project to ask, could this actually be useful as a way to monitor patients in the clinic? And it turns out that it probably can. So here's kind of a cheat experiment. It's really obvious, but it does show that it can work at some level. So this is a patient who had surgery. We'll hear about this exact thing later. So you had an IDH mutant tumor. Here's 2-HG. You get surgery. The tumor is removed. The 2-HG is gone. We took away the tumor, the 2-HG is gone. More importantly, if you were treated with some drug, so this is just standard of care, okay? So this is a patient treated with radiation and standard of care chemotherapy who did well with it, and you can see that this also, you can read out the improvement
by decrease in 2-HG. And one thing that's really cool in a paper that was a collaborative paper with this group at MGH that was just submitted argues that how well the patients do using just sort of standard metrics of how well patients do can really be also predicted by this response. And so I think this is pretty exciting because it says that, well, as you'll hear from Kate, maybe we can target this for better therapy, but also it says that this is some way that we can go through and actually monitor these patients and know if, we'll hear from Steve later, if he does ever need to go in a direction where he would require some of these drugs, we can know if they're working perhaps earlier without having to be really invasive. And I want to save time for you to hear others, so I'll just stop now and thank the, first of all, I can't ever give a talk and not thank the awesome people in my lab. I'm so fortunate to be here at MIT and work with really amazing people. And I've tried to mention some of the other folks who were involved in this. And again, I just want to highlight again the Bridge project, which has funded some of our lab work around IDH mutant cancers, as well as, you know, some of the great core facilities and other support that we've gotten here at the Koch Institute. So thank you. I think we're going to move directly to Kate, right?